Hello friends, I am Lakshmi Kant. Hope you all are fine, my dear. Here in this very topic, I am going to start Kingdom Monera, my dear. So in previous part, that is in part 1, I was told you about the classification, how many types of classifications, as well as the scientists who tell about the different types of classification. The first scientist might be know that very name I was told in previous lecture, that is Aristotle, right? And then after the two kingdom classification was given by the scientist whom you know, that is Carlos Linnaeus, that is father of taxonomy, my dear. And then after the Ernest Haeckel gave three kingdom classification, four kingdom classification was given by the scientist Copeland, and the fifth kingdom of classification was given by the scientist R.H. Whittaker in 1969. Correct? And then after the one classification is also, that is sixth kingdom classification, that is kingdom Monera, further be divided into two parts. In kingdom Monera, you already know that the sole organisms, that is bacteria. Bacteria is further divided into two parts, that is archaebacteria and eubacteria, right? So this one is classification is called sixth kingdom classification, and this kingdom classification was given by the scientist Carl Woese, my dear, right? Okay. So now here, what here we should have to know here, the first classification, that is kingdom Monera, my dear. So in this very part 2, totally I discuss about the kingdom Monera, that is sole organisms, that is bacteria, my dear. Okay, so now we start the kingdom Monera. Okay, let me start the kingdom, which one? Kingdom Monera, my dear. So I'm writing here what? The kingdom Monera, my dear, right? Kingdom Monera. In kingdom Monera, what we know, the sole member here, that is bacteria. Here the sole member, which one? Here bacteria, right? Is that clear? Okay, so in kingdom Monera, here bacteria is what you already know, that it is cosmopolitan. It is what? Cosmopolitan, my dear. So if I'm saying here that word cosmopolitan, it means what, you know, that is found everywhere, my dear. Okay, so bacteria, the habitat is what? Each and every place. So that's why I'm writing here in a first point, that is what? Bacteria is a sole member of kingdom Monera. So what I'm writing here, bacteria is sole member. See what I'm writing here, sole member of kingdom, which one? Kingdom Monera. What the meaning of sole member? अकेला, है ना? तो किंगडम मॉन्ड्रा के अंदर बैक्टीरिया क्या आ जाता है? सबसे सोल मेंबर आता है, यानी सबसे अकेला यही मेंबर है। जब हम लोग बात करते हैं बैक्टीरिया के बारे में, वी ऑलरेडी नो दैट नंबर, यदि हम लोग नंबर की बात करें बैक्टीरिया का, तो नंबर क्या है बेटा? आप जानते हो, लार्जेस्ट नंबर। व्हाट आई एम सेइंग? लार्जेस्ट number of what? Microbe, my dear. What I'm saying here, microbe. Microbe means what? The organisms which cannot be seen with our naked eyes. So largest in number. Okay. Now next thing is here, habitat. What I'm writing here, habitat. See, if I'm saying here habitat, so where it found? It found where? In a soil, in air, in water. So that's why I'm saying here what? Cosmopolitan. What I'm writing here? Cosmopolitan. So what the meaning of cosmopolitan? That the meaning is what? Found everywhere. Each and every places the bacteria found. Right? Now, here the habitat, that is unusual habitat. Unusual habitat means what? The habitat where we can't believe that the place the bacteria are alive. Yes. So see, unusual habitat. What I'm writing here? Unusual habitat. See, if I'm writing here unusual habitat, then where bacterially it is found in acidic medium, as well as what I'm writing here, high temperature, clear, as well as what, deep sea my dear, गहरे समुद्र में भी मिलेगा, deep sea also, even a stomach of ruminant animals, that is herbivorous animal, stomach of which one animal, ruminant, ruminant animal, correct, correct. So universal habitat is what, acidic, it is found in acid also, high temperature also, deep sea also, stomach and ruminant animal also. Okay, so now see, I'm saying here about the bacteria. Here we further classify this one bacteria into two parts. In previous part, I was also told about the bacteria further classified into two parts. That is what, archaebacteria. One is what, archaebacteria, and second one is what, eubacteria. Second one, which one, eubacteria. Clear? So here the bacteria further be classified into how many parts? Two. One is what, archae, second one is what, eubacteria. Now see, if I am saying here about this one is the bacteria, see, the bacteria is cosmopolitan, number one. Number one, wait for a moment. Here the kingdom monera, my dear, correct? What I am telling here about kingdom monera, that is kingdom monera means what, bacteria, it is the sole member, अकेला प्राणी है ना kingdom monera में पाया जाने वाला क्या है, yes, that is bacteria. And here the bacteria which is found what, everywhere, so that's why I'm writing here word what, cosmopolitan, right? Bacteria is the sole member of kingdom monera. This point is clear. Largest number of microbes, habitat every places, either the soil, air, water, so that's why I'm written in bracket cosmopolitan. Unusual habitat also bacteria found, that is in acidic medium, high temperature also, deep sea also, stomach of a ruminant animal also. Clear? Now the bacteria is what, two. When we classify the bacteria into, one is what, archaebacteria, second one is what, eubacteria, my dear. See, after knowing these all things, now I'm going to classify the bacteria. Now see, here I'm saying what, classification of bacteria. See what I'm saying here, classification of what, my dear, classification of bacteria. See, whenever I'm going to classify the bacteria, now see how we classify. Number one, on the basis of, see what I'm writing here, on the basis of shape, my dear, correct? On the basis of shape, number one, that is what, coccus bacteria. What I'm writing here, coccus. So coccus, which one shape? That is a spherical in shape, like one. This one is coccus bacteria. Now, second one is what, bacillus. What I'm writing here? I'm writing here what, bacillus. That is what, a rod-like structure. That is what, bacillus, correct? Bacillus. Third one is what, that is vibrio, my dear. Okay, vibrio. If I'm saying about the vibrio, then which one? Let's see, vibrio bacteria in this way. That is, you say what, comma say bacteria in this way, the shape, not in this way, my dear. Wait for a moment. In this way, that is a comma shape bacteria. Clear? Vibrio. Now next one, that is spirillum. That is what, Spirillum, spirillum bacteria. So how the spirillum bacteria, let's see. This one is this one is a spiral bacteria. Okay, so coccus is what singular, cocci word is what plural, bacillus is singular, bacilli is what plural, vibro is what singular, vibram is what plural. Okay, clear, clear. So most common shape, that is what most common shape. Let's see what I'm writing here. Wait for a moment. Most common shape, I'm going to write here what most common shape. What the most common shape, my dear? See, the most common shape is what? Bacillus. What I'm writing here? Bacillus. And second thing, that is what? Coccus, lack of flagella. Coccus, lack of what? Flagella, my dear. Correct? Correct. So this one is the important thing about the kingdom Monera, that is what? Bacteria, my dear. Okay. So now see the NCRT line. Here, what I tell you here, see, see. So bacteria are the sole member of kingdom Monera. This line I have written in the previous slide. The most abundant microorganisms. Most abundant means what? It found in large amount. So bacteria occur almost everywhere. Just I told about this one thing. Hundreds of bacteria present in the handful of soil. Handful mitti mein hundreds millions of bacteria. They also live in extreme habitat. What the extreme habitat? That is what? Hot springs, deserts, snow, deep oceans, where very few others life forms can survive. In that very place, the bacteria can also live. So many of them live in or other organisms as what? Parasites. Some are what? Parasites also. Clear? This thing is clear, my dear? Yes or no? Tell me first. Okay. So now, then after, see what I'm going to tell. Now, here I'm going to tell you about some NCRT line. That is what? NCRT line here. See what I'm going to write here. What I'm going to write here. Bacteria, bacterial structure is very simple. Bacterial structure is what I'm saying, very simple. See, if I'm saying here that the bacterial structure is what? Very simple, they are complex in behavior. Very simple. They are what? They are what? Complex. Complex in what? Behavior. See what I'm saying. The structure is what? Very simple. Bacterial structure is what? Very simple, but they are very complex in behavior. This one is the NCRT line. So here they show the what? Most extreme metabolic activities. See, bacteria as a group, so the most extensive metabolic diversity. Next point I'm writing here, what? That is, for a moment, that is bacteria as a group, because they are found in a group. So the most, so the, what I'm saying, most extensive metabolic, what? Metabolic what? Diversity. See, metabolic diversity. See what I'm saying. Bacteria as a group show the what? Most metabolic diversity. So if I'm saying here that the bacteria is what? Most metabolic activity, it means what? They have different metabolic characteristics also. Okay. See how I'm saying this one thing, because they synthesize their own food. I am saying this one thing because they synthesize their what, my dear? Own food. So how they synthesize their own food, my dear? From the inorganic substances. Or you say the from inorganic substrate. What I'm saying? Inorganic substrates. See, here I'm saying that whatever the bacteria, they can synthesize their own food from inorganic substrates. So they may be photosynthetic, autotrophic, or you say the word chemosynthetic. So what I'm writing here in the next point, they may be what? They may be photosynthetic. See what I'm saying here? Photosynthetic. So see, if I'm saying here that the photosynthetic, autotrophic, photosynthetic what? Autotrophic, as well as, or you say the chemosynthetic, or you say what? Chemo chemosynthetic, my dear. See, so they may be photosynthetic, or you say the word chemosynthetic, because autotrophic of two types. One is photosynthetic, second one is what? Chemosynthetic. So see, the bacteria is mostly either they having autotrophic photosynthetic, or you say the chemosynthetic. Now, next thing that is important here, that majority of bacteria are what? Heterotrophs. Majority, some weird word, photosynthetic, but majority of bacteria is what? Heterotrophic. So that's why I'm writing here majority of bacteria are what? Are what? Heterotrophic, my dear. What I'm saying? Heterotrophic. Clear? That is, they depend on the other organisms. Heterotrophic, that is what? Depend on what? Other organisms. See what I'm saying here? Depend where? On the other organisms. So majority of the bacteria are heterotrophic, but some are photosynthetic also. Either they having photosynthetic or the chemosynthetic. So uh, majority of the bacteria is what? Heterotrophic, which depend on other organisms. Clear? Or you say in heterotrophic, or you say on dead organic matter, or on what I'm writing here? Dead organic matter. Clear, my dear? For what? For food, my dear. So this one is the clear, clear. C and CRT line. So this one is the shape of the bacteria. See, this one is the cocci bacteria. See, this one is what? Cocci bacteria, which is spherical in shape. This one shape is, this one is spherical. This one is a spherical shape. Bacilli word is plural. This one is rod shape bacteria. Clear? This one is the spore. Either they also found what? A spore, a spirilla. Clear? This one is the vibrio, spirillum, having this one is the flagellum. Okay, and vibrio having what? Comma shape bacteria. Comma shape bacteria. Clear? So now here I'm going to make the diagram of the structure of bacteria so that it will give more clear about this one. Okay. So see, I'm going to draw a diagram of what? Bacteria. See how I'm going to draw. Let's see in the next slide, my dear. See in this one. I'm going to draw a bacteria. See, bacteria has three-layered membrane. Clear? Wait for a moment. First I draw, then I level the bacteria. Here, bacteria has three-layered membrane. Yes, bacteria has how many layered membrane, my dear? I'm telling three-layered membrane. This one is the what? Outer membrane called glycocalyx. Clear? See, I'm going to write the name. This one is what, my dear? This one is glycocalyx. See what I'm writing here. This one is glycocalyx. Clear? Now, in white. But this one name, this one is called after glycocalyx. This one is what? Cell wall. This one is what? Cell wall. Okay. And now the inner membrane. This one is what? This one is called cell membrane. This one is called cell membrane. Together called, but I'm saying this together known as what I'm writing here, cell envelope. What I'm writing here, cell envelope, my dear. Correct? Now see what I'm going to draw here. Here the cell membrane having inner folds. This is called mesosome. This one is what? Mesosome. Clear? This one is what? Nucleoid. See, this one is what? This one is plasmid. This one is what? Plasmid. See, this one is what? This one is plasmid. Clear? This one is what? Flagella. This one is fine. Here like structures known as what? Fimbriae. This one is what? Pili. What I am taking. This one is what? Inner membrane having inner folds. This one is called chromatophore. This one is what? Chromato. Let's see. This one is what? Ribosomes. This one is what? Ribosome. This one is what? Chromatophore. Wait for a moment. Chromatophore. These are called what? Mesosome. This one is what? Flagella. Clear? This one is called fimbriae. This one is called pili. Okay. So now inside the flagella there is a rod-like structure. Clear? This rod-like structure is called filament. This one is what? This one is filament, and this one is what? Nucleoid. This one is nucleoid. Clear? Now this one is what? Here. This one is called inclusion bodies. Clear? See what I'm writing here. This one is what? Inclusion bodies. Clear? So this one is the diagram of what? Bacteria. So each and everything here I'm leveled. That is what? The glycocalyx outer layer. Number first. This one is the second, cell wall, and third one is what? Cell membrane. This glycocalyx, cell wall, and cell membrane together known as what? Cell envelope. Correct? So this one is the plasmid. This one is the flagella. Clear? And see, in flagella, here divided into three parts. This is called basal body. Basal body. This one is called hook. This one is called what? Hook. And this one is what? Filament. This one is what? Filament. Clear? Clear? Yes. So these all are the diagram of what? Bacteria. Clear? Now see from NCERT. See what the things I was told in previous slide. This one is written in your NCERT also, my dear. See what? Though bacterial structure is what? Very simple, but they are very complex. So this statement I was also written in previous slide also. See this one statement I am written here. See, bacterial structure is what? Very simple. I am written here na? Very simple I am written here. See, very simple, but they are complex in what? Behavior. See, I am written here, so that's why I'm telling you, check here. Things is also written in your NCERT. So simple in structure, but complex in what? Behavior. See, compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. This one is also given by me also. Some bacteria are what? autotrophic. What I'm saying, some are autotrophic, that they synthesize their own food from inorganic substances, yeah, inorganic substrates. They may be photosynthetic if they have photosynthetic autotrophic, or you say the word the chemosynthetic autotrophic. Majority of the bacteria are what? Heterotrophs. This one told. They depend on other organisms or eat and decompose organic matter. Clear, my dear? So this one is the point I was told in previous slide also. Now see. Next thing. That what the important. So here I am going to write what? The structure of bacteria. Clear? So with the help of diagram I must shown here. Let's see the diagram. This one is the diagram of bacteria. See, this one is the structure of bacteria. So now I'm going to proceed each and every structure of bacteria in details, my dear. Clear? Now see in this one. ठीक है? तो स्ट्रक्चर ऑफ बैक्टीरिया में हम लोग क्या देखेंगे? सबसे पहला बात हम लोग किसकी बात करेंगे? स्ट्रक्चर ऑफ बैक्टीरिया में किसकी बात करेंगे? फर्स्ट हम लोग बात करेंगे किसका? सेल इनवलप का. सबसे पहला क्या था? सेल इनवलप. सो हियर वी टॉक अबाउट विच वन? सेल envelope, my dear. So cell envelope to get which one? That part, that is outer region glycocalyx, then after the cell wall, then after the cell membrane. So cell membrane is the innermost layer of the bacteria, outer membrane is glycocalyx, in middle there is what? Cell wall. Clear? So cell envelope किसको किसको मिलाकर हमने बोला था? याद करके देखिए. सबसे पहला क्या था? आउटर. आउटर क्या था? चेक करे थे डायग्राम में. ग्लाइकोकैलिक्स क्या था? ग्लाइकोकैलिक्स, राइट. फिर इसके बाद सेकेंड मिडिल में क्या था? मिडिल लेयर क्या था? कमेंट सेक्शन में आप बता सकते हैं डियर. मिडिल लेयर में क्या था? यस, दैट इज सेल वॉल. व्हाट आई एम सेइंग? सेल वॉल. और अंदर का जो लेयर था बेटा, थर्ड इनर मोस्ट लेयर is what? That is, you say what? Cell membrane. That is say what? Cell membrane. Together known as what? Cell envelope. Together known as what? Cell envelope, my dear. So this one is given page number NCERT of 127 and 128. Clear? You can check also NCERT page number. NCERT page number. You can also check 127 and 128, my dear. Correct? See, here I am going to tell you about the outer layer. Number one, that is first glycocalyx, that is outer layer, my dear. Correct? So see what I am saying here, outer layer. Here the outermost layer of the bacteria, which is divided into two parts. Number one, that is what? Slime layer. Slime layer. And second one is what? Capsule. If they having a capsule, then it's called capsulated, otherwise not capsule. Correct? So now see, here the slime layer are of two way, that is either they have loose and slimy. What I'm saying here, loose and slimy I am saying. And second one is what? Second one, that is you say thin layer. In layer, clear? Okay. Now in a capsulated, if they have, then it is what? Then it has what? Thick layer. Then it is thick as well as what? Tough. And which one strain is here? S strain. Which one bacteria is here? S strain. And if they having S strain, then it have what? Capsulated. Then it has what? Capsulated. Correct? Correct. So if they are having what? S strain, then it means what? It's make what? Capsulated. Right? So glycocalyx is the outer layer of the bacteria. Okay. Now second layer that is cell wall. Second layer that is what? Cell wall, right? So now I am saying about the cell wall. Then what I am saying here about the cell wall? See, this one is what? Plant. You know that the plant is in plant made up of what? Cellulose. But if the fungi also having cell wall, that is called chitin. Then I'm saying here what? Chitin. So now if I'm saying about the bacteria, in a bacteria they also having a cell wall. So in a bacteria it is made up of carbohydrate along with the what? Amino acid. Along with the what? Amino acid. Okay, that is called peptidoglycan, right? Clear? So this one is the structure. Now see here, whatever the things that is archaebacteria. So when we move through the archaebacteria, the first of all we should must have to know about the what? Cell envelope first, then after we have to be move ahead. Okay, in the archaebacteria and eubacteria. So first of all I would like to tell you about which one. Wait a moment, I'm going to add a slide, so that's why I'm taking here. See, this one is thing is clear. So before we move to this one archaebacteria, here we need some more extra slides, so that's why I will explain here all the different parts of what? Yeah. See what I'm going to tell you in this slide. See, in this slide, what I'm going to tell. Let's see. Now I'm going to tell you about on the basis of cell envelope. See what I'm going to write. This color, my dear, I want to take color white. Okay. Yes, take color. Which one? White is visible to all of you. See what I'm writing here. I'm going to write here on the basis of what? Cell envelope. On the basis of what? Cell envelope. The thickness. On the basis of cell envelope, the thickness is be differ. Yes or no? So on the basis of thickness of cell envelope, number one, and second thing on the basis of thickness of cell wall. See what I'm saying here. I'm saying here, dear, here we classify the bacteria in two ways. On the first, on the basis of the cell envelope, then after on the basis of the thickness of cell wall. Correct? See, when I'm going to tell here on the basis of thickness of cell wall, then we classified into two parts. One is what? One is gram positive bacteria. One is what? Gram positive bacteria, and second one is what? Gram negative bacteria. Correct? One is what? Gram positive bacteria. Second one is what? Gram negative bacteria. See what the difference between gram positive and gram negative bacteria. See, my dear, first point here. In a gram positive bacteria, cell wall is what? Thick, my dear. Correct? While in gram negative bacteria, cell wall is what? Thin, my dear. Correct? Number one. Now, number two, here retain stain. It means what? Color. retain a stain, my dear, but in gram-negative bacteria do not retain a stain. Do not, what I'm saying, retain stain, my dear. This is the second difference. Now third difference is what? Here the example of that is what? Lactobacillus is a gram positive bacteria. Lactobacillus as well as Clostridium as well as what? Clostridium. And in this one, here the bacteria that is what? Rhizobium. Rhizobium is gram negative bacteria. Rhizobium, or you say the E. coli bacteria is also. Cyanobacteria also. Cyanobacteria, as well as you say the mycoplasma. Mycoplasma, these all having what? A gram negative bacteria. Now here in a gram positive bacteria, they are containing what? Mesosome. So see, mesosome is here, while in this here what present? Here porins present, that is opening porins, villi present, and this one is what? And toxin. So that's why I'm saying what? This one is PPT. Clear? This thing is clear. So now see. So if I'm saying here that which one is the gram staining bacteria, and gram staining bacteria was given by the scientist gram staining bacteria was given by the scientist Christian Gram. Which one is the scientist? Christian Gram. Correct? Correct. Now next, on the basis of now the cell membrane, other part is what? Cell membrane of bacteria. See here the cell membrane. But the cell membrane, we all know that the cell membrane is made up of lipid bilayer. What I'm writing here in cell chapter, that is in cytology, you already study about the cell membrane. The cell membrane is what? Lipid bilayer. What am I saying? Lipid bilayer. See, if I'm saying here the lipid bilayer, it means what? It is made up of phospholipid. It is made up of what? Phospholipid. See the structure of this lipid bilayer. I'm going to draw. This one is what? Lipid. This one is what? Lipid bilayer. So above upper end is called hydrophilic. Upper end is what? That is polar head. Hydrophilic end, and this one is what? Hydrophobic. This one is what? Hydrophobic, my dear. Correct? Correct. Now see here what the important point to be know here. Here the very important point to know here, my dear. What the important point here? Let's see. Plasma membrane is structurally similar. Here important point is what? Plasma membrane, that is also called cell membrane. Plasma membrane. Plasma membrane, at what I'm saying here, plasma membrane are structurally similar. Are structurally, what I'm saying, similar to whom? To eukaryotic cell. to eukaryotic to eukaryotics. See, so if anybody can ask you about what the plasma membrane, the plasma membrane are structurally similar to eukaryotic cell. So whatever the eukaryotic cell, the plasma membrane is the same as the plasma membrane also found in which where in a bacteria also. Correct? Now see. Next point, that is what mesosome. See something about what mesosome. Here, the extension of plasma membrane. You know the extension of plasma membrane. I will make the diagram there also. See what I'm writing here. Extension of what I'm writing here. Extension of what I'm writing here. Plasma membrane. See here I'm writing what extension of plasma membrane. Yes or no? So extension of plasma membrane. So here the diagram, that is outer membrane glycocalyx. This one is the cell wall, and inner membrane having inner extension. This one is called mesosome. Right? This one is what? Mesosome. Yes or no? Because this outer layer is called glycocalyx. Glycocalyx. This one is called which one? This one is called cell wall, and inner membrane is what? Cell membrane. This one diagram, na? Cell membrane is made up of what? Lipid bilayer, phospholipid. So now the extension of plasma membrane. See, this one is the extension of plasma membrane known as what? Mesosome. Known as what? Mesosome. So see here, extension of the plasma membrane on the basis of surface area. What I'm saying here? On the basis of what? Surface area. It classify into three parts. It classify into how many parts, my dear? Into three parts. So see what I'm saying here. I'm saying here that the extension of plasma membrane is called mesosome. So mesosomes can classify into three parts on the basis of the word surface area. So see, and this one is the diagram to make. So suppose number one, that is vesicles. So if this one diagram is like this, then it is called what? Vesicle. Okay. Now, second one, if the structure is like this mesosome, then it's called tubular. Now third one is like in this way, then it is called lamella. Then it's called what? Lamella or lamellae. So there are three types of mesosome. One is vesicles, second one is tubular, and third one is what? Lamellae. So this one is the what? The different types of mesosomes, which is the inner extension of the plasma membrane. Okay. Now see what the role here. What I'm going to write, what role? See, role of mesosome. See here, enzyme for respiration. Wait a moment. Number one, enzyme for respiration. Enzyme for what? Enzyme for respiration. So see, if I'm making a diagram here, the outer layer glycocalyx, . Outer layer is what? Glycocalyx. Second one layer is what? Cell wall. And this one is the third layer called mesosome. And this mesosome having inner fold. See, this one is what? Mesosome. So this mesosome is responsible for enzyme for respiration. Clear? Second one is what? Cell wall formation. Cell wall formation. Now, third one is what? DNA replication. When the DNA replicate, this one is the mesosome. DNA replication. Then after fourth point here, that is what? Cell division. Cell division. Here the major four roles of mesosomes. One is what? Enzyme for respiration. Second one is cell wall formation. Third, DNA replication. And fourth one is what? Cell division, my dear. Correct? So now here some bacteria also contain what? Chromatophore. Am I right? Diagram I was already made in previous slide. See here the chromatophore. Chromatophore is only present in blue-green bacteria. Chromatophore. It is a pigment that are attached. Where it is present? See here what I am writing here. This one is the what? Chromatophore is what? Pigment are attached on the membrane. Attached on the where? On the membrane known as what? Chromatophore. See, so if I make this one diagram earlier, just make there. See, this one is the glycocalyx. This one is the what? Glycocalyx. This one is what? Cell wall. And here the inner membrane, that is what? Plasma membrane, or you say the cell membrane. Clear? So this inner membrane having inner folds. This inner fold is called chromatophore. And this chromatophore containing a what? Pigments which are attached on the membrane known as what? Chromatophore. And this can only be present where? This one is glycocalyx. This one is what? Cell wall. Right? And this one is what? This one is called cell membrane, my dear. Correct? So together known as what? Cell envelope. So here I am saying what? This extension of the word cell membrane from what? Chromato. From what? Chromatophore, my dear, which is basically found where? In a blue-green algae. In a blue-green cyanobacteria. That is what? So, you say, my dear, that is, you say what? Cyanobacteria. Sorry. Not blue-green algae. You say what? It is present where? This one is what? Cyanobacteria. Correct? Now, the other thing, that is also some flagella. See, these all thing I was made in the diagram also. See, this one is the diagram. This one is the diagram. To tell you, this one is the diagram. Clear? See, here the mesosome term is clear. Mesosome term is clear. Chromatophore is clear. Okay. Now I'm going to tell you about what flagella. This one, flagella. See in this slide, mesosome is clear, chromatophore is clear. Now I'm going to tell you about what flagella. So write down the point. That is what flagella, my dear. So what about the flagella? See here the structure. Here the basal body. Inside the basal body, this is the what hook, and this one is what flagella. Clear? So see, this one is basal body. This one is what hook, and this one is what? This one is what flagella. Inside the flagella, there is a rod-like structure known as what filament, which gives shape. Is this one is what filament. Okay, so if anybody can ask you about the flagella is made up of what? Flagella is made up of what? Protein. What I'm saying? Protein. So which one protein? Here the flagellin protein. The name of protein? The name of that very protein is what? You say flagellin protein. What I'm saying? Flagellin protein. So this flagellin protein having different types of protein. Having what? Different types of protein, my dear. Correct? Correct? Now, if anybody can ask you about this one is the prokaryotic, na? This one is a prokaryotic, but eukaryotic flagella, eukaryotic flagella is made up of tubulin protein. Is made up of what? Tubulin. So see, what the thing is important here to know, that is what structure of flagella having three parts. That is what? See, number one, basal body. Number two, hook. Number third is what? Filament. And this one is what? Flagella. So it is made up of what? Protein, flagellin protein. And this flagellin protein, which one? Prokaryotic. This one is what? Prokaryotic. So prokaryotic flagella is made up of what? Flagellin, which having different types of protein. While in eukaryotic, the flagella is just made up of what? Tubulin protein. So this one is very important to know. Clear? Clear here? Now, next thing is here. In note, you can write down non-motile flagella. Non-motile bacteria, you say. Non-motile bacteria doesn't contain flagella. Doesn't contain flagella. What I'm saying here? So if I'm saying here that this one is the point is very notable point that you have to be keep in mind. So whenever I'm saying that, so non-motile bacteria, then that bacteria doesn't contain what? Flagella. So if I'm saying that the bacteria containing flagella, it means what? They having what? Motile bacteria. Okay. So now see, non-motile bacteria doesn't contain flagella. Here is some example also. Which one example here? Nostoc. Nostoc you say. Anabena you also write down. Nostoc, not Anabena. Focus you say. Focus as well as what? Mycoplasma. They do not contain what? Flagella. Clear? Clear? Now see, other structure, that is what? Pili. Other structure, that is what? Pili. So this one is the structure of pili, na? Suppose this one is the structure of what? This one is what? Pili in a bacteria, right? So if I'm saying about the pili, that is what? It is tubular in shape. What I'm writing here? This one is what? Tubular in shape. What the shape? Tubular in shape. Less number. Less number, but large in size. In size, my dear. Clear? Clear? So now see. So if I am, this one is the pili. Now I'm going to tell you about the what? Fimbriae. So see. I'm drawing what? Fimbriae. Fimbriae. So see the structure of fimbriae. This one, bacteria suppose, and this one is what? Find here like structures. This one is what? Bristle shape. This one is what? Bristle shape. This bristle shape, more in number, more in number, and what? Small in size. Small in size. So if anybody can ask you what the difference between pili and fimbriae, then what you say? In pili, tubular in shape, while in a fimbriae, bristle in shape. In pili, less number but large in size, while in fimbriae, more in number but small in size. So this one is the difference between the what? Pili and fimbriae material. Correct? Hope so clear? Hope so clear? Now, here, one thing that is also very important to note, that what? Here, what I'm writing? Pili plus fimbriae. What I'm writing here? Fimbriae plus flagella. Flagella together called what? Surface structure. Together called what? Surface structure. Correct? So this one is clear? Clear or not? Tell me first. Okay. So these all are the what? Some parts of flagella. Clear? So now in next part, my dear, some little bit parts also left here. So in next part, I'm continuing. That is the next part. That is what? Nucleoid, and then after the plasmid, types of plasmid, and then after the what? How many types of plasmid there, and ribosomes. There's some ribosomes is also some little bit point is be left there. So in next part we will discuss that. Okay. Thank you for watching. Thank you.